Okay, hello everyone. Welcome to today's webinar on extracellular vesicles isolation with HansaBioMed using TFF technology. And I'm Tina, the product manager from Rocker. And it's a pleasure to have you all here with us. We all know that extracellular vesicles plays a crucial role in intercellular communication, which is associated with both physiological and pathological processes. And HansaBioMed is dedicated to the research of extracellular vesicles and is a pioneer in this field. So today we are very delighted to invite Triin from HansaBioMed to share her valuable insights into the benefits of TFF in the EV purification. So now let's welcome Triin.
Okay, thank you, Tina, for this introduction. And hello, everybody. My name is Triin, and today I will speak about how tangential flow filters can be really beneficial in the exosome vesicle research. Before we go to the topic, I would like to say a few words about our company, HansaBioMed Life Sciences, which really was established more than 15 years ago in Estonia, with an exclusive idea to dedicate to the research and product development in the exosome and extracellular vesicle area. And because we were really the very first company to do that, we have become today the pioneering company in the EV field. In our everyday life, our company have many different international research projects going on. We have many important collaborations from where we really strive the innovation to the EV field. And besides tangential flow filters that we sell in our catalog, we also have different products like purified extracellular vesicles, fluorescent vesicles, different EV-specific isolation tools, characterization kits, DNA, RNA isolation kits, and very good, well-validated EV-specific antibodies. And besides the products, we also perform custom services to our customers to meet the need of our customers. And what are the extracellular vesicles? So basically, these are membrane-surrounded nanoscale structures that can be released to the extracellular space by all types of cells. They carry cell-specific information like different proteins in them and nucleic acids that can be really beneficial to the extracellular vesicle and prognostic and diagnostic tools. And because the vesicles carry EV cell-specific cargo, as I mentioned, they can be really beneficial in the diagnostic and prognostics. And there are several subtypes of vesicles. The very first class is called the exosomes, or the small EVs, that appear in the extracellular space through multivesicular bodies. So when the multivesicular bodies fuse with the plasma membrane, they release its content via exocytosis, and these little bubbles that you see on the screen, they become the exosomes. And the second is called the microvesicles, or the large EVs, that has a defined size from 150 to 1000 nanometers. And these are generated by budding off the plasma membrane. And also when the cells go through apoptotic bodies, these bodies are discharged by cells that has a variability in the size from 100 to 5000 nanometers. And recent findings also talk about exomers or supermers that have distinctive morphologic characteristics to them. And the main and most fundamental issue throughout EV research has been how to isolate the vesicles. And EVs can be purified from various biofluids, or like urine, serum, plasma, They can be isolated from bacteria, from all types of plants, and also from cell culture conditioned media. Before the purification can take place, this biomatrixes have to be eliminated from various contaminants like cells and debris. And then the enrichment can be performed by different methods, depending on the isolation volume and experimental aim. And the very first way and most conservative way to purify EVs is the ultracentrifugation, which is based on the high sedimentation force to pellet defined size of particles. Then immunoaffinity is based on the antibody-antigen capture efficiency. It is favorable towards smaller volumes, and isolation of different subpopulations of vesicles is achieved with the highest purity, but with a moderate yield. And then size exclusion chromatography is based on the gravity flow and pore size in the agarose matrix. Polymer-based precipitation is a really cost-effective way to obtain vesicles that has decreased purity index. And most importantly, the tangential flow filtration, when it's combined with size exclusion chromatography, has become the most favorable and mainstream isolation method in the extracellular vesicle purification. And in tangential flow filtration, the fluid is passing parallel to the filter rather than it's being pushed through. The particles that pass through the membrane, which are mostly proteins and different smaller molecules, are put off to the side, while the rest, the retentate, is recycled back to the feed. And this method is really preferred for its continuous filtration. It is favorable when the biofluid has really concentrated filtrate matter, and it is also because there is a constant turbulent flow across the membrane. It prevents the accumulation of cake formation on the membrane surface. But with the dead end filtration, the situation is a little bit different because the complete feed flow is forced through the membrane, and the filtered matter is accumulated on the surface of the membrane. That can lead to pore blockages and cake formation. And in time, there is also the penetration of the membrane, penetration rate of the membrane will also decrease with time. And this also has a negative effect on the yield because the particles stick together to each other and block the membrane surface. And I would like to also bring out another important aspect, that tangential flow filters can be really well washed out and used multiple times. So really it's a cost-effective way to do the purification, while with the dead end filtration and centrifugation of concentrators, because due to the pore blockages, they must be discarded. Because the ultracentrifugation was the so-called gold standard in the EV purification in the past, and tangential flow filtration, when it's combined with size chromatography, is becoming a new gold standard. I would like to just highlight the aspects of these two methods. And in ultracentrifugation, we are going to pellet the vesicles with a high sedimentation force, which in most cases is more than 100,000 G. It results in a sticky vesicle pellet that can also have a fractioned vesicle membrane because of the high sedimentation force. And when it happens, the vesicles release its precious cargo. While in tangential flow filtration, it doesn't happen because there is a smooth flow of particles across the hollow fibers and EVs do not integrate. In ultracentrifugation, the recovery and purification yield is rather poor, and there are numerous studies showing that you need to process at least 10 times flasks to obtain the same amount of vesicles, while with TFF you can do with it one. So this is really becoming also an expensive way to cultivate the cells and do the EV purification. Ultracentrifugation is rather time-consuming. The minimum time it requires is 4 hours. There is also capacity to the volumes that you can purify, while in the TFF there is continuous flow that can be regulated even up to 120 milliliters a minute, depending on the viscosity of the fluid. Then ultracentrifugation does not really fit for very small volumes, because you do not obtain any kind of you would obtain a very small amount of vesicles, but either it's not suitable for large scale because due to the capacity limitation that the rotor has. But with the TFF, it can be scaled from 5 milliliters up to liters and thousands of liters, basically, if you change the filter. There is also, in ultracentrifugation, there is this contamination problem, because whatever you have in the sample, it will pellet. It is contaminating proteins, for instance, albumin that comes from fetal bovine serum. It can be these smaller vesicles that I mentioned, these exomers, supermers. It can be lipoproteins, and it really does not it gives this contamination issue. But in tangential flow filtration, you can actually fractionate your sample depending what kind of pore size you're using. You can also take several pore size filters and fraction the samples to your requirements. And this really can eliminate those contaminating subpopulation of the vesicles like exomers, supermers, microvesicles, and so on. So the ultracentrifugation is also expensive, rather expensive equipment, and requires us a specially trained technician. While with the TFF can be used from HansaBioMed, that this can be used manually. It can be connected with the syringes, or for large-scale production, you can connect it easily with a peristaltic pump that has really not as expensive as the ultracentrifuge. And another aspect that in future, when we want to approach the diagnostics from humans, we need to purify clinical grade vesicles, and tangential flow filters are sterile equipment. So the purification can be performed under the hood to obtain those sterile vesicles that can be used in the cell culture studies as well. And the majority of publications to the EV research actually arrive through in vitro cell cultivated and purified vesicles. And fetal bovine serum is one of the most common supplements for cell growth. But it is evident that fetal bovine serum actually contains various exogenous vesicles that have a bovine origin. And those vesicles can be taken up by nearby cells and also modulate the function of the cells and cellular signaling. So if those vesicles are not eliminated from the preparations, they also will be isolated with sample of interest. And I would like to show how the TFF-EVs filter from HansaBioMed is really easy and efficient way to deplete EVs from fetal bovine serum. And we have done this experiment working under the safety cabinet, keeping the cellular growth and cellular viability and preserving the sterility. And in the study, we have taken 10 milliliters of raw fetal bovine serum. We have processed this serum through TFF-EVs filter. all of the vesicles coming from fetal bovine are retained inside the filter. And further on, we have analyzed the retendate and also the filtrate with nano-tracking analysis experiment, and further we have separated the particles by size exclusion and chromatography to further estimate the efficiency of this method. In order to understand how the cells grow in different conditions, we have used 3 types of cells. We have conditioned them in without serum. We have conditioned them with FPS depleted medium and with a complete medium. And we have monitored the cell viability within 3 days. And in these results, you can see that the TFF-EVs cartridge was basically able to eliminate majority of the vesicles from FPS. The retendate, the filtrate contained only very little amount of the particles. And we also did fractionation of the nanoparticle from raw FPS and from the filtrate. And you can see that the filtrate, which can be now used for cells, contains less than 1% of the nanoparticles inside, with isolated with size exclusion chromatography. And it's very important to understand how the cells grow. So we have monitored different conditions, and from these charts you can show that all of the cell lines, the cell growth even expands on the 3rd day, so it equalizes and has really positive effect on cellular growth. And to conclude, the TFF-EVs can be used for depleting fetal bovine serum or other cell culture supplements from nanoparticles, including vesicles. This operation can be run under the safety cabinet, preserving the cell and medium sterility. The filtration through the TFF-EVs filter removes 99% of the total bovine serum nanoparticles, which will be retained inside the filter. And the use of nanoparticles depleted FPS does not impact the cell viability. Another important or application where the TFF can be used is when researchers work, for instance, with urine vesicles. Those who know that in urine, the majority contaminating protein is this Tamm Horsfall protein or uromodulin, that basically forms a network that entraps the vesicles, and those, when you do the purification, you will obtain a very little amount of vesicles because the majority of them will be purified out with Tamm Horsfall protein. And to have a solution to this problem, the main issue would be use some chemical reactions like TPT or CHAPS to raise the solubility of the uromodulin. But we want to show that the TFF is really efficient and versatile way to eliminate this Tamm Horsfall protein, contaminating protein, in a short time and with an extremely simple, scalable, and reproducible procedure. And in this study, we have used 40 milliliters of urine. We have processed it through TFF-MV filter cartridge that has pore size of 200 nanometers. We have collected, this is the retendate, and we have further taken the filtrate and processed it through the TFF-EVs. Now we are going to purify the small exosomes or small EVs. And in the TFF-EVs procedure, we do the washing step to wash the vesicles well, and then we do the recovery of the vesicles in 4 milliliters of PBS. And in the particle analysis study with NTA, we can see that about 15% of the vesicles, which are the large-scale vesicles, were retained inside the filter. We further processed the total of 65% of the flow-through, and we obtained around, from that we obtained 22% of the urine vesicles. So overall it would be 60% of the total particles for the of the urine vesicles. That actually is a very good recovery for urine. And we also wanted to assess the protein content in this purification process, and we see that the flow through of the MV filter contains about 80% of the proteins, and the purified vesicles would be like around 5%, meaning that we were able to eliminate in the flow through of the TFF-EVs about 99% of contaminating proteins. And it's very important to understand what are these proteins and do we see the expression of these EV-specific markers. And indeed, we see that in a total urine, we don't observe any kind of marker because there is a dilution factor so big. But we see that TFF-MV retentate, this 200 nanometer pore size filter contains a lot of Tamm Horsfall protein. So this is very helpful to eliminate this protein. And we see that the purified urine vesicles with the TFF-EVs cartridge have a very nice exosome specific ALIX marker on them. While it's absent from the flow-through, which contain other proteins. And we see also very good tetraspanin expression on the urine vesicles. It is very present CD9, CD63, and CD81, and also the size distribution is very beautiful. And to conclude, the double tangential flow filtration is a simple and fast protocol which can be carried out manually on the lab bench or under the safety cabinet, providing high EV yield and purity. There is about 60% of recovery of the total particles. You can remove contaminating proteins, including Tamm Horsfall protein. There is a turnaround time less than 10 minutes, and it's very easy because you do not have to pre-treat urine with some chemical. And our company really has several different types of tangential flow filters in the catalog, although they appear in the same physical appearance. They are really different in the characteristics. So the very first I'm going to talk about is the TFF-MV. It aims large vesicles or microvesicles, and with this filter you can separate, for instance, small vesicles from the large vesicles. The cutoff value is around 150 nanometers, so all the particles bigger than this number will be retained inside the filters, and when you collect the flow through, you will receive the small vesicles with additional proteins. This cartridge is made of hollow fibers of polyether sulfone. It is sterile. The cutoff value is around 1000 kilodalton. This you can process with this type of filter up to 2 liters of volume. This mainly depends the viscosity of the biomatrix. Since, for instance, as we all know that serum and plasma are more thick than urine or cell culture media fluids. And our company really has TFF is unique in that context that if you have very small volumes to purify, you can simply connect this cartridge to the syringes and you can process them as manually or mechanically. And we have also several video guidance on YouTube where you can see how actually to work with these cartridges. and that's why we are the only provider of such TFF for manual use. And then there is TFF-Easy cartridge. This has a pore size 5 nanometers and a cutoff around 15 kilodaltons. So it is mostly a concentrator because it retains everything inside the filter, and basically some very small proteins and water is passed through the membrane. And this can be used for different biofluids, plant extracts, cell culture media, and EV dialysis. It is also sterile and can be processed up to 2 liters of volume. And as I mentioned before, EVs, TFFs cartridges are very good to fraction different size of molecules. So if you want to work with the small EVs like exomers, then this cartridge is perfect. And then there is a third type of filter. This is called TFF-EVs, has extracellular vesicles or exosome, and this has a pore size of 50 nanometers that retains particles from that size and bigger. In here we can offer 2 different filter typologies. We have the small one and the large-scale production EV purification. And with the bigger one, you can process material up to 10 liter. The TFF is really fast and rapid way for EV purification. It takes less than 5 minutes. It can be, as I mentioned, used under the hood and for sterile cell culture work. You can process volumes small or big, and there is also very big interest of academic teams. And why TFF is beneficial? And first of all, it is a very gentle method for EV concentration and purification compared to dead-end filtration. You will obtain very high recovery of vesicles. No EV aggregates are formed because media does not become sticky during processing. You can remove various proteins and ions during filtration. You can combine it with size exclusion chromatography to upgrade the purity of the vesicles, but it can be also simple concentration after the size exclusion chromatography purification. The cartridges are washable, reusable multiple times, and they are also sterile. And there are different applications for TFF. You can perform dialysis with that cartridge or dissolve your sample. You can isolate nanoparticles, all types of nanoparticles, extracellular vesicles or viruses. You can remove from your sample bacteria and endotoxins. You can clarify cell lysates with this method. You can fractionate biomolecules. You can harvest cell suspension. You can work under the hood and produce sterile vesicles and use it for sterile cell culture work. You can eliminate unwanted proteins from the sample, for instance, as I mentioned, from Tamm Horsfall protein from urine, and you can deplete vesicles from fetal bovine serum. And Tina is going to show you a video on how to use the TFF with the filtration system.
HansaBioMed TFF Hollow Fiber Cartridge, Quick Start Guide. Remove the screw caps. Install an empty syringe onto one port and a syringe with sample onto another port. The filtrate port can be connected to a proper container or drained directly. Confirm the direction of the three-way stopcock on both sides. Push 2 syringes alternatively upwards and downwards until the desired volume is reached. The filtrates will be collected into the container while concentrating. Close the filtrate port with screw cap. Push the concentrated retentate to syringe A. Remove the screw cap on the other side. Adjust the three-way stopcock and load air with syringe B. Adjust the three-way stopcock again. Push the air into cartridge to collect the remaining sample with syringe A. Adjust the three-way stopcock and remove the syringe A. Concentration is complete. Fill the syringe with millicue water, then install it onto the cartridge. Confirm the direction of the three-way stopcock on both sides. Push two syringes alternatively upwards and downwards to wash the cartridge. The TFF cartridge can be reused after cleaning. If not used immediately, please dry and store the cartridge at room temperature. Install the cartridge onto Tanfil 100 TFF system. Start concentration. Concentration is complete when the sample reaches the desired volume.
So thanks, Triin, for such insightful presentation. And it's fascinating to explore the potential of TFF and combine with the SEC as a new gold standard in the EV isolation. So now let's open the floor for questions, and please type your question in the chat box. So, Triin, in the like small EV research or so-called exosome research, what suggestion did you have for using your TFF cartridge? You will recommend to use two different cartridges or to use only one cartridge and then followed by SEC?
Actually, yes, it's true that before you can work with exosome preparation, you need to eliminate large vesicles from the preparation. And for that, HansaBioMed has actually two different types of TFFs. We have the TFF-MV filter, in which you can eliminate all of those large vesicles from the sample. And when you go collect the flow through, you can do purification of the small vesicles. Alternative for the TFF to eliminate these large vesicles from the sample is to perform high-speed centrifugation. But with that, there can be also contamination because you never will obtain all of the vesicles with centrifugation. And secondly, yes, that can affect this preparation. While this TFF is a good way to fractionate all the biomolecules, that also has increased recovery.
Okay.
Yes.
And in your presentation, you have mentioned that the old gold standard, like the ultracentrifugation, they proceed the sample in the non-sterile condition, and TFF can do it in the sterile condition, and it is washable and reusable. So
yes,
people are curious about how to ensure the sterility of cartridge when reusing it.
Yes. Actually, it's a very good question because indeed the TFF is sterile when it comes in a package. When you open it under the hood and work with cells, then it is sterile. But if you are going to reuse it multiple times, then of course the sterility is gone. And in that case, when you need to work with sterile conditions, I always recommend taking for instance, more material to process in one go and then later discard the filter, because the filter cannot be autoclaved, but it can be re-sterilized only with gamma irradiation if it's possible. Otherwise, if it is important to keep the sterility, then yes, I cannot guarantee to to have it sterile over half or 6 months, because it's true. But our purification protocol has really good cleaning steps that comes with a heavy and easy fouling. So you can use detergents to eliminate all particles inside the hollow fibers, because the polyether sulfone fibers, they are very resistant to low and high pH shifts, also different temperatures. So you do not have to be afraid to break the polyether sulfone fibers in that case. You can really wash it well and reuse it next time. You can be sure that all the particles have been washed out from the filter.
Okay.
Yes.
So in this case, if the users want to make sure the sterility, you will recommend to enlarge their samples, like to 5 liter or more than
for instance,
after one time use.
Yes, for instance, if the aim is to keep the sterility, as I mentioned, it can be used to eliminate exogenous vesicles from fetal bovine serum. You can process, for instance, more 1 liter in one go than aliquot the sample and freeze it put it in the freezer. And in that way, you have used the maximum of this filter and also kept the sterility. Okay. There is not any other possible way.
Okay,
yes.
Thanks. And the other question is about, you say the new gold standard is the TFF and combined with the SEC, right? And
yes.
My question is, I use the TFF to remove the large particles, large vesicles, and then concentrate to the amount of quantity, and then put it into the SEC, right? And then did you compare about I just used the TFF as the purification technique, or to combine them together?
Yes. Actually, the TFF-EVs filter can be purification method on its own, because the majority, it has a pore size 15 nanometers and the cutoff value around 900 kilodalton or 850. So the majority of the proteins actually go to the permeate. But if you need to really upgrade the purity really to the finest level, then yes, some researchers perform next step with size exclusion chromatography, which is also very doesn't require a lot of time, like 10 minutes maybe, to do it. But it's not necessary, because the EVs recovered from the filter can be already analyzed for Western blot, for an NTA. It is really absolutely fine. Yes.
Okay, so there's no more questions. Okay, so I think today's webinar is just to hear. So it's time for us to close the meeting. So thanks again for Triin for this excellent presentation. And thank you all for your attention. So before we officially close this meeting, I'd like to ask you to take a moment to fill out the certification surveys, and your feedbacks will help us to enhance the future webinar. So see you next time. Bye-bye.
Thank you. Bye.